So unlike most humans, I try to focus now to the present. Since I have to give a lecture, maybe I will think about the next slide. But before I start, I would like to make two remarks. One of them, I'm really honored to be invited to this symposium, and thank you very much for the organizer. And the second thing is that I can't stop admiring the organizers who basically launched this entire National Academy of Science education. I just would like to know for the students that it is indeed one of the best things which could happen to you in the past couple of years, that you have the chance to participate in this academia. And I'm really glad that it went so well and so many talented students turns up, and at least we have some chance that some of you will continue and participate in research. So congratulations for all the organizer. Maybe one more remark. We started this symposium with cancer. Okay, then we continued with Alzheimer's disease. Now comes stress. So I don't really want you to be depressed that life is so sad and we all have these maladies. So actually, being in the lab is a big fun. You discover something every year and every day, actually. And it's really motivating. But we face many challenges in life. And as a scientist, we need to solve many of them. And now we will focus on stress. So it is a little bit sad, but it turns out that none of us is an exception. And during our lifetime, we will certainly experience traumatic events. And if we are lucky, if it comes only once. It can be any type which are listed here. Accidents, violence, sudden death of a loved one, or war, or surviving a brain injury. So it can come in many times, but there is one very important thing, that a single event, like a stress, can have enduring effect on your behavior, which lasts long, long time, in certain cases for your entire life. And this has been recognized, and you probably know that these are called stress disorder, and I'm pretty sure that all of you know this term PTSD, which is the post-traumatic stress disorder. It's much less known that there is an acute form of this disease, and this is called acute stress disorder. And the difference in the diagnosis is simply between the time of diagnosis. From 3 to 30 days, you call it acute stress disorder. From the 31st day, you call it post-traumatic stress disorder. What's more, ASD can actually quite frequently, up to 70% of the cases, turn into PTSD. What's more, the treatment of ESD is actually much more easy than the treatment of PTSD. So if you try to interfere early in this process, you may have a better chance to cure the patient. As I mentioned, the symptoms of ASD and PTSD are very similar. We cluster them into symptom cluster, like intrusion, negative mood, avoidance, arousal, and stress. Intrusion is the negative memories. Negative mood is obviously depression, avoidance, that you withdraw from interacting with your environment. And arousal and stress is a general term. It refers to problems falling asleep, hypervigilance, so too much activity, and intense emotional distress. Now, where and what changes in the brain during this process is actually pretty much unknown. For one reason or another, huge amount of work have been put into revealing the mechanism of PTSD, but actually we know very little what happens in your brain right after the stress hits you, and what may be altered, which may cause the development of acute stress disorder. So let's see now the brain. This is sagittal sections of a mouse brain, the outlines. This would be the cortical regions, or where the neocortex is resigned, and this would be the subcortical centers. It is pretty much known that when stress hits our brain, it first reaches the subcortical centers, most notably the hypothalamus, which obviously launches the hypothalamic pituitary stress circuit. So basically, these subcortical centers set a stress state. It can be more stress, less stress, so it's a dynamic state. However, the stress-related behaviors are actually organized by cortical regions. So these are higher order brain centers, like the prefrontal cortex, hippocampus, and amygdala, and these will determine all these phenotypes or behaviors which I refer to as stress phenotype. So the question is, what links these two? And there is a peculiar situation that there is a hub in the brain which actually receives all these subcortical information and projects all to these cortical centers which organize the behavior, and this hub is called the paraventricular nucleus of the thalamus. I will refer to them as PVT. Interestingly, this is the only subcortical region which has fast and direct access simultaneously to all these three brain centers. There is no other. There is only one day. So it's really a bottleneck between the communication of subcortical and cortical centers. The transmitter of this nucleus is glutamate, and this is the fastest, most precise, and most effective transmitter in the brain. The neurons of this paraventricular nucleus in the thalamus can be labeled by a calcium-binding protein called carotinin. Obviously, you may ask, what is the situation in the human brain? This is the position of the thalamus in your brain, and if you make a section in it, then you can see that the position of PVT is actually in the very same position than in mice. So this would be the human thalamus. This is the lateral, this is the medial part, and this actually in the very core, the most medial part of the thalamus is where you can find PVT. Obviously, you can appreciate how giant, large, or cortical regions are compared to thalamus, and not to speak of the PVT. So it's not immediately logical why such a small region would be responsible for this very widespread projection, but apparently this is the case. So if we make an immunostaining for carotinin in a human section, and we zoom in this image, or at least try to zoom in, try again, like a monkey. Okay. Then what we see that indeed at the cellular level, dark spots appear which are carotinin positive interneurons, indicating that indeed we have the same type of neurons than the mice. Moreover, if we stain the inputs, in this case we stain the hypothalamic inputs, these black, fine thread-like structures are actually axons coming from the hypothalamus, and you can appreciate how specifically they innervate the brown carotinin cells. In these regions, which is the neighboring thalamic regions, you don't see any fibers, so indeed this hypothalamic information is funneled to these cells not to these other neurons, which are not labeled but keratin. And astonishingly, exactly the same thing happens in the rat. So this is the human brain. This is actually, that is mice brain. Again, you have the carotene positive neurons, hypothalamic inputs, highly selective, no carotene cells, no input. So we can confidently say that PEV is highly conserved between mice and man, and this obviously has translational relevance, since we hope that what we find in case of a rodent might be applicable to primates or humans. But I will ask you to give me 2 minutes to this word conserved. We frequently say that during evolution one structure is conserved or not. I would like to have a brief tour that when we say so, what we actually mean. And for this, we should make a little bit of time tour. And for this, I will use this very intelligent software called Earth-Viewer. It's worth to download it from the HHMI homepage. So this is obviously our Earth. Here you can see the time. And we jump back to 65 million years ago, OK? You can still clearly recognize North America, South America. And with the help of this software, I can show you the position of huge impacts, meteorite or asteroid impacts on the Earth. And what we are most interested in now is the little bit difficult to pronounce Kix Club, whatever, impact crater in the just south to North America. And this is the Cretaceous tertiary impact, which happened exactly 65 million years ago. And as you all know, this is the one which killed all the dinosaurs. Now, I think just last year in Montana, which is actually not too far from the impact, discovered the remnants of a very nice, cute animals. Obviously, these are reconstructions based on the bones, and he's called Purgatorius. And his age is 64.9 million years, a mere 100,000 years after the impact. So we can pretty sure that this guy survived the impact, right? Because there is no way that such an animal evolves. So Purgatorius probably lived through this huge impact, but there is another even more interesting thing. Is basically he is the first primate or proto-primate. He still has a mouse or rodent-like look, but actually he already is an adept climber, which is a primate feature. So we can pretty sure that he is one of our ancestors. So how we can link these three animals, the mouse, man, and the Purgatorius? And actually now we are pretty sure that the mammalian branch, which includes both rodents and us, actually were split pretty early. And it can lead to creatures which, at least in two cases, we know the organization of the paraventricular thalamus is very similar. And as an evolutionary thinking, it should mean that the ancestor, the common ancestor of this branch, also should have a very similar organization. So basically, we know that the KT boundary is somewhere here. So we can firmly conclude that this is a very, very ancient structure. And when we mean that something is shared between mouse and man, we mean that we are talking about a feature which is present in these creatures for longer than 600 or 65 million years. So obviously, the question is that if this structure has very unique and selective inputs which confer stress, if it projects to all the brain centers which are involved in organizing stress behavior, and we know that it's present for at least 60 million years in our brain, then can it take part in establishing ASD-like symptoms? So we put a very simple hypothesis that the activity, and specifically the activity after the stress, is necessary to establish these ASD-like symptoms. The question is that how can you test it? Fortunately, there is a big breakthrough in neuroscience that you can selectively interfere with the activity of neurons using a magic method called optogenetics. You basically enter opsins, light-sensitive opsins, into the membrane of the neurons, and if you put a thin fiber optic next to them and shine light to it, their activity could be significantly really diminished, but just during those time when you switch on the laser. So basically, it's highly specific temporally, and we can actually inhibit only the activity of carotenoid cells, nothing else. So using this tool, we set up the following experimental procedure. First, we had the mouse in their home cage and examined their behavior using a camera, their sleeping using EEG and movement EMG, and we also measured the neuronal activity of the PVT neurons themselves. Then we exposed them to their natural predator, but we don't have a fox in the lab, so we bought in the shop fox urine odor. And we took the animals to a different cage and exposed them to fox odor for 10 minutes. So this was the stress. This was the stress situation. They are really stressed, completely stressed. It's an innate fear. Obviously, they never met a fox, but it's an inherited fear of the smell, so they are really frozen like hell. Then we put them back to their home cage, and then came the magic, the optogenetic inhibition. So we inhibited the activity of PVT neurons, but only for 1 hour. Obviously, we are scientists, so we needed a control, and the control is, in this case, an uninhibited cells. These animals receive the same treatment, so laser, everything, but the neuronal activity in PVT is unaltered. And then we tested the animals for another 4 days, and this would be the post-stress days, referring to the ASD. We measured the following things. We measured hypervigilance, but how much they move, logical. The sleep disturbance, how much time it takes to fall asleep. And the intense emotional distress, that how much time they spend freezing in their nest. Probably you heard this term freezing. This is when you basically hardly move, just basically your heads and not, we are really frightened. And obviously, we also measured how the PVT neuron firing is altered. This is what we found. Oh, obviously, we always compared the pre-stress period to the post-stress period. So this is what we found. First, I show you the comparison of the control animals, pre-stress versus post-stress. This would be the hypervigilance or the movement, simply how much they move. And indeed, you can see that this is time 0 when you start the experiment, that after the stress, the animals indeed move significantly more than before the stress. So indeed, the stress evoked hypervigilance. They move actually just by looking. They are really stressed, and their movement is really different after the stress than before the stress. If we measure the freezing, indeed, after the stress, they spend really a lot of time freezing in their nest. So it's really you can say that it's an intense emotional distress, and it took them significantly more time to fall asleep. Each dot in this case is an individual animal, so you can see the variance of the effect, but you can see that, let's say, this animal froze almost 3 times more after the stress than before. So basically, we can see that the three core symptoms of ESD-like symptom have been presented by these animals. And now comes the critical experiment that we inhibit the animals, but just once, and we measure the effect later. So in case of movement, we could actually reverse the effect. So the animal stopped to be hypervigilant, they behaved normal, or even moved a little less than before the stress. The distress completely were abolished by a single exposure to the inhibition, so they simply freezing time were unchanged, and similar sleep disturbance disappeared. There were no significant difference between the pre-stress and post-stress rate, so we conclude that the inhibition works. Single inhibition or 1 hour inhibition of the structure after the stress basically blocked the development of ESD-like phenotype. What happens with the neurons? So this is the activity of a PVT neuron before the stress and after the stress. These are actually action potentials, what the neurons generate. And this is the mean firing rate, what we call how much they fire. And actually, this is the same neuron. You are able to record one neuron for several days. So these PVT neurons, as you can see, actually tripled the firing rate after the stress. So they went to another state where their average neuronal activity was pretty much higher. And actually, in every state we tested, there was a very, very dramatic increase in firing rate. And most interestingly, what we found that before the stress, these would be the gray columns, these individual dots are actually individual neurons. So you can see there is a huge variability in firing rates. And this is the average which goes up after the stress. But you can see that before the stress, there is a very nice state dependency in the firing rate. So when the animals are awake, it's pretty high, and as they enter the nest, they start to go down, and when they fall asleep, it falls down again. So there's a very nice tendency. I'm getting to be sleepy, going tired, want to sleep, and then it's very low when you sleep. And this is what is especially screwed up, and especially the largest trait is in the nest. So basically, the animals enter the nest, and instead of feeling, oh, calm down, I'm going to sleep, they still are stressed, they are freezing, and their neuronal activity is pretty high. So paralleling the change of the behavioral observation, we see an increased PVT firing rate. Let's see how inhibition affects this. And to our big surprise and satisfaction, the change, the significant increase of firing rates induced by the stress could be completely eliminated by the inhibition. So basically, not only the behavior, but the firing activity of the PVT cells remained normal just after this one treatment. So as a conclusion, we can say that our principal idea is that the stress hits the brain, and basically its activity travels through the PVT. It alters its activity, and it sets into a new mode. And this bombardment of altered activity basically rewire the cortical centers, and they will form a stressed circuit. And this stressed circuit will be responsible for the development of ESD-like phenotype. Now, if we block this activity right after the stress, and we do not allow this altered activity to develop and bombard the frontal cortex, then the ESD-like phenotype does not develop. So we can clearly see that the post-stress activity of PVT carotene-positive cells are necessary to establish the ESD. And Presently, obviously, we do it with photoinhibition, but obviously you can't really use it in case of humans. But the idea that once you find a selective intervention which would target these carotenoid cells, we can hope that there will be a drug which could ameliorate the adverse effect of stress behavior. And like all the others, I would thank for the group who have to bring forward, as well as for the grant support, mainly Anna Jász, László Bíró, and Zsolt Budai. And I'm pretty sorry, but Kata Kóta is not shown here. She is our student, and we are pretty much happy that this academy provided a talented student for us. So thank you very much for your attention.
Thank you very much, Professor Acsády. We are waiting for questions from the audience, maybe 1 or 2 short questions. We still have time for that, and then we close the session. There is maybe somebody over there, so please. Yes, I can see you.
I would like to. Sorry.
Okay, I'm sorry, just doesn't work. So I would like to check on the beginning of your fascinating presentation, and I would like to ask you 2 quick questions. One of them regards to ASD. What makes some people more sensitive to stress than others? Because as far as I'm understood, ASD might develop into PTSD, but it does not happen in every single case. So that would be one question of mine. The other one is about the mice. So as you mentioned, they were exposed to fox otter. But if you were to show them a video of fox, for example, for 10 minutes as well, would that make them as stressed as the other made them? Thank you.
Okay, thank you very much. Very good question. The first one is obviously the $1,000,000 question, and this relates to vulnerability, as we call it. Certainly, I draw your attention that the amount of response, behavior response we measured were highly variable from animals to animals, okay? So indeed, we could see that the impact of stress is different from animal to animal. This we can also detect in case of ASD. In case of PTSD, actually the vulnerability of the population, we know it from the human population, is even lower, is somewhere between 20 and 30%. So only 20 to 30% of the humans will develop PTSD after this strong stressful event. And presently, there are lots of research to figure out what makes you sensitive for PTSD, and whether would it be possible to characterize your behavior or genetic or whatever background which would predict that once such a thing happened, then PTSD likely develop. Obviously, once we had these measures, then it would be much easier that bad things happen. First you have ASD, then PTSD. Then if you belong to this vulnerable population, immediately add some drug and try to avoid the PTSD. Concerning the second, mice are not visual animals. So actually, as Purgatorius, as all these other guys, evolved to fill a night niche. Maybe because of the dinosaur ruled the Earth during the daylight, maybe for other reasons, but basically mammalians are pretty much hooked up with living night and resting on tactile information, auditory information, and odor information, but not vision. So primates are actually the most visual mammals, and with this, we actually reduce both our nasal as well as our auditory sensory experiences. So that's why I'm pretty sure that, I don't know, do you have a cat or dog at home? I mean, you can show whatever you want in the cat, in the TV, even the most, they maybe try to catch it once or twice, but cats will give up to hunt birds in the TV because they rely on smell, touch, and hearing, similar to the mice. Thank you for the question.
Maybe for a last short question. Yes.
So thank you for your brilliant presentation. I have only a brief question. How did you measure the neuronal activity?
Okay, so these are very fine wires, 10 micron each, okay? So your hair is something like 70 or 80 microns, so it's really much thinner than hair. You bundle them together, you lower it, and there is a trick here because he wanted to measure carotene neurons. So these carotene neurons were actually labeled by an optogenetic tag, and with the wires we also put down a fiber optics. And once we shine a light to the fiber optic, then the neuron start to fire the action potential. So then we knew that these are carotene cells. So the trick about recording neuronal activity is that during the action potential, there is an exchange of sodium and potassium ions, and as a consequence, in the extracellular milieu, there is a change of these ions. So you can record them as a change in the voltage, and you amplify it, and this is the way you record neural activity.
Stunning. Thank you very much.
Okay.
Thank you very much. Let us hold this certificate and give it to you. Thank you for the kind words at the beginning of the presentation.